Hello there, this is Dr. Dan Guerra, and today is the 22nd day of September, and the year is 2021. That makes it the first day of autumn. I prefer the word autumn to fall, I don't know why. And I'm really happy because autumn is my absolute favorite season. So, if I could be playing music right now, I haven't quite figured out how to do that without it sounding like I'm playing it from a speaker. I would be playing Vivaldi's Autumn, right? The third concerto in the Four Seasons. And maybe I'll play it later on my Facebook page just to remind you that that's what I said was thinking about, and maybe what I was going to do, if I had a better audio to do that. But what we're going to do today, besides think about Vivaldi in the fall, that is the autumn, is we're going to continue our discussion of the synoptic lectures involving aging. So I'm going to call this Aging Synoptic 2. It's the 22nd September, and again, Dr. Dan Guerra. Now, that image you see on my front plate there is not me, although it might look a little bit like me. That is my dear father, and I think that was probably taken right after World War II in Chicago. And I haven't quite figured out how to look at what's on those signs, but I need to do that. The signs behind him, because I think that would tell us something about the date and maybe just about what was going on. This is probably like 1945, 46, maybe even 47. I don't really know. But I know I inherited that old black and white picture, and that is him. So let's get right into the lecture. He obviously was aging well at the time. He was in his 20s, and that's a big difference to what I am. And it's a big difference to what he finally became, which was much older before he passed away. So we've been talking about this aging process from the point of view of biochemistry and physiology. We've talked a lot about cell metabolism, a lot about whole organism alterations in the aging process. And I've done this now for months, and so I plan on consolidating that. This is part 2 of the video lecture. So let's just go ahead and get after that. For some reason, there we go. So first of all, let's do this distinction of terms. I like to do that before I delve into descriptions. And we've talked about this on the audio lecture, so I'm almost repeating myself. But apoptosis, programmed cell death, sensu stricto, requires particular cysteine-dependent, aspartate-specific protease. Those are called caspases for short. And they produce an orchestral disassembly of the cell as they cleave cellular substrates, resulting in cytoplasmic and then finally nuclear condensation. DNA cleavage and maintenance of an intact plasma membrane is essential for apoptosis, as it does not induce a pro-inflammatory response. The degradation products of apoptotic cells are packaged into membrane-enclosed bodies, and finally they are targeted for phagocytosis and removal in vivo, resulting in a complete absence, or some would say a great diminishment of inflammation, which is what's necessary. This is a cell fate that requires no further damage be done to the rest of the system. Now, uniquely, caspase-1 dependent programmed cell death there's another type of it, and it's called pyroptosis. This is a programmed cell death, and it's stimulated by a range of microbial infections. For example, these bacteria, Salmonella, Francisella, and Legionella. And then non-infectious stimuli, including host factors produced during, for example, a myocardial infarction, wherein caspase-1 processes the inactive precursors of interleukin-1 beta and interleukin-18 into their mature form. It's a proteolytic processing, obviously, because caspase is a protease. And then you get rapid cell death, characterized by a plasma membrane rupture before this compartmentalization, and that releases pro-inflammatory intracellular contents to the extracellular space. And then you are going to get a bona fide inflammatory response from both the innate and ultimately, typically, also the acquired immune system cellular pathways. And we talked a lot about this, so we don't need to give you an extra reference now. Let's get into some detail here. A paper published in Cells in 2020 gives us a little bit of insight what might be going on intracellularly with and without an apoptotic fate. Short hairpin RNA for an enzyme called acid sphingomyelinase can be responsible for perturbing homeostasis of what's known as the cholesterol and ceramide level in a lipid raft. There's a ratio of ceramide, which is a kind of a sphingolipid we talked a lot about here. And the ratio of ceramide to cholesterol alters not just the composition of a membrane lipid raft, it also alters its fluid density and therefore mobility in the cell. So that lipid raft and the endoplasmic reticulum itself leads to an alteration of a protein called GRP78. And that protein gets membrane trafficked and subsequently induces a P85 alpha unphosphorylated P10 complex. Now, P10 is typically a phosphatase, not a kinase. So you have an unphosphorylated P10 complex with that P85 as induced by this lipid raft through GRP78. So that caused a disruption of the canonical pathway we've been talking about in lecture. The phosphatidylinositol 1,3 kinase, the AKT, and the GTP-RAC1 mediated signaling, and the ER membrane-associated GRP78 typically regulated oxidative stress balance. Thus, all of that will inhibit cancer cell invasion. Now, what this suggests, this one paper with this data that we just basically summarized for you, not showing you the evidence, but that's what the evidence shows, is that ceramide, via that acid sphingomyelinase that we just inhibited with a short hairpin RNA, which is basically a pharmaceutical, that the ceramide promotes metastasis via raft biochemistry. We can now call it pathobiochemistry, which is going to involve the P13 kinase pathway. That's what you get from that kind of information. Okay, so that means you can turn that switch off and on, and that means the switch is readily available. It's readily available no matter what the cell lineage might be, as long as all those genes are expressed. Now, this means that the membrane dynamics alone can regulate cell fate. What kind of cell fate? The two we just described, for example, apoptosis, ferroptosis. And we also know about autophagy. Right? Of course we do. So let's continue on here. Now, within autophagy, and we're going to go into more detail what I mean by that, but obviously autophagy is a reorganization of the cell. It's not going to lead to a terminal death. Okay? Reorganization, basically to allow the cell to withstand whatever the vagaries of stress were that were targeted to it. Now, within autophagy, you also have organelles that can be auto-digested through the phagolysosome. And one of those organelles is the peroxisome. We talked about the peroxisome quite a bit in this class, I'm calling it a class, in these series of lectures. And so we're going to go back to it, but I just want to show you that studying yeast gives you a lot of similar ultimate fate of a peroxisome mechanistic evidence as does studying it in mammals. Is that always the case? Oh no. But in this instance, the protein players are different, but the organizational degradation of the peroxisome is similar. The reason I'm bringing this up is sometimes it's good and useful to use model organisms. Yeast is a model organism. So you can see here, let's just look at the mammalian one. There are proteins called PEXs, and PEX2, PEX3 will induce this intermediate phase of the peroxisome, which becomes ubiquitinated. Okay? And that whole system can be embedded into an autophagolysosome by increasing these proteins in their expression. And you can see what they are, the ATG8PLC3, I guess it's LC2, that's what the green protein is there. You also, again, have this peroxin protein, and you have the ATG receptor. All of that has to be in place. You get P62, you get this NBR1 protein, and you get this protein number 14, is what it's called. And ultimately, you generate the scaffolding that's going to eliminate the peroxisome. Now, why would a cell eliminate the peroxisome? An entire organelle involved in oxidative metabolism, involved in membrane turnover and resynthesis, plasma membrane in particular. It's also involved in the elongation and desaturation of some of those very long-chain polyunsaturated fatty acids we've been talking about, such as those from the omega-6, omega-3 series. Okay, so why would you eliminate a peroxisome? Well, peroxisomes age. And so an autophagy allows you to take a peroxisome out of commission in a cell, have peroxisomal genesis then, and that peroxisomal genesis will allow you to maintain the cell once the older peroxisomes, which have somehow lost their utility and therefore function, have been eliminated. So this is a whole other thing that goes on in a cell. Autophagy associated with organellar degradation. And phagy just means to consume, right? So here you can see the autophagolysosome is involved in removing peroxisomes. Same thing happens with mitochondria. Now, interesting thing that most people might not know, but that you know, because I'm telling you, and I've told you before, mitochondria and peroxisomes can actually replicate themselves. They don't involve only what the cell is doing in terms of cell cycle. You can make multiple mitochondria and multiple peroxisomes within one cell. And they replicate according to a fission-fusion phenomena, which I know I talked about way back, probably now 6-7 months ago. I'm not going to go back and talk about all that subcellular dedifferentiation and then differentiation. But just keep in mind, the peroxisomes and mitochondria are two subcellular organelles that can actually duplicate without the rest of the cell duplicating itself. So you could have multiple peroxisomes, multiple mitochondria. Now, why would a cell do that? So that it can carry out the functions of the peroxisome and the mitochondria at a higher or lower level of degree. You see. So embedded within the cellular response that you think about when you think about autophagy, as I say here, is specific turnover of the peroxisome. And that's termed peroxophagy. Now, since peroxisomes can occur in multi-structural motifs, including the various stages, so peroxisomes can exist in 6 different stages, so there are proteins associated with the stages, and those are the PEX proteins. They're characterized by a developmental differentiation in protein quality and quantity, as well as size of the peroxisome, and the subcellular distribution of the proteins and lipids that I described elsewhere, but also must assemble. So you have to have specific peroxisomal membrane lipids, and you have to have proteins that are associated with those lipids, because the peroxisome has this membrane around it, and it has to be able to carry out the functions of a peroxisome, which means it has to control communication. Now, one structure, termed the peroxisomal reticulum, not to be confused with the endoplasmic reticulum, is particularly important in certain mammalian tissues and cells, such as the sebaceous glands, where it is often confused with the ER under microscopy. Closer inspection reveals a unique lipid and protein profile. Regardless of the structure, the dynamic scope of protein and lipid translocation and turnover within the peroxisome clearly distinguish it from the ER, the Golgi, and of course, the mitochondria. So this is all really relevant. Now, why am I bringing it up? These are generic concepts. This isn't even really biochemistry, pathophysiology. But you can see that all of this internal structure goes through a turnover. So for the cell to maintain its integrity, which basically means it carries out the same function it was first evolved to generate or produce, those organelles themselves, within those cells, have to turn over. If they don't turn over, the cell loses its homotypic functionality. If that occurs, the cell is basically no longer a dynamic and healthy system to maintain in a tissue. So it must be destroyed. But as long as it can go through autophagy, it can maintain its existence, you see? So autophagy is a way to prevent terminal cell fate, which is death. Now, this is sort of a microcosm of what goes on. Okay, that's a subcellular microcosm that goes on with all the cells. Ultimately, their fate is to maintain and divide until they reach a certain limit. They stop dividing because of mutations and epistatic interactions, and then epigenetic alterations, which are always happening. Remember, writing into the epigenome, reading of the epigenome, altering gene expression, and then erasure of the epigenome. Then starting over, starting with a new chalkboard, or completely stopping epigenomics altogether and letting that cell just simply divide, or ultimately letting that cell senesce or necrose, depending on signaling and then internal phenomena. Okay. So the cells and then the whole organism ultimately follows suit when there's enough repair, enough recombination, enough alteration of generations of proteins from early, middle, to late in the sequence of the cellular life. When that gets tuned up to the point where there are too many mutations, too many epigenetic cross-references, if you want to look at them that way, the whole organism starts to become unhealthy. And this ultimately leads to a falling down. The curtain is coming down on the whole organism. And these are things we normally recognize as morbidities. Those morbidities ultimately lead to mortality. Right? So that's, again, going all the way from the very micro to the macro. And that's what I like to do here, is to keep you kind of aware where we're going. So I don't want to lose you with the granularity. Okay? So as with pexophagy, as a recurrent turnover mechanism for peroxisome-mediated removal of oxidized membrane lipids, both development and aging are under the aegis of a hormonal influence, diet, nutrition that is, exercise, and of course intellectual endeavor. But aging cannot be attenuated while physiological development relies on set points and endpoints that reach equipoise at more or less defined temporal benchmarks, more or less. So aging results ultimately in cellular senescence and loss of recovery after stress, as the immune system becomes both highly informed through such cellular and biochemical mechanisms like T and B cell memory generation, and of course epigenetic alterations of toll-like receptors, microRNA expressions is now all epigenetics, and covalent modification of chromatin, which is more to do simply with biochemical phenomena, not anything related to an epigenetic phenomena, just the trafficking and interaction of communicational systems at the biochemical level within each cell lineage. And these are going to be prone to error as you age. They're going to be prone to error because of things like oxygen, too much oxygen. You have too much reactive oxygen being generated just from normal bioenergetics, for example. And because of that, you make more and more mutations, and that can lead to more and more cell lineages that are no longer functioning correctly. And they must be removed or disposed of. So beyond autophagy to apoptosis. And sometimes that apoptosis, which is clean and neat, can be wiped. The slate can be wiped clean actually with macrophages, like type 2 macrophages working with type 2 T helper cells, right? And we talked about that in one of the audio lectures recently. Even that whole system breaks down. And ultimately, you have to start dealing with degeneration. When enough of the cells degenerate, the tissue is no longer functional, and then goes an organ. And then when the organ starts to degenerate, then you get into heavy-duty morbidity, like liver damage, lung damage, kidney damage, cardiac damage, or just endothelial damage, or CNS damage. And then an entire plenum of that degeneration will lead to mortality, you see. So the individual is unique both biochemically and epigenetically. So each individual organism is unique because all these systems do not change according to a Newtonian mechanistic universe understanding. They change somewhat stochastically. That means somewhat randomly. And because of all the possible permutations, certainly into the hundreds of billions, into the trillion of phenomena, individual phenomena, any given cell lineage, because of all the interactions of the cells with other cells, I'm sure that number is appropriate, trillion. Ultimately, there are too many decaying, shall we call them, cellular hypotheses, right? Which lead to cellular hypostases and a destruction of the entire oosia, which means the organism in this particular contemplative metaphor, and you lose then the integrity and the organism dies, the human dies, right? So individuals unique both biochemically and epigenetically, even while inherited components, sensu stricto again, set a range of possibilities. I don't like the word probability because that sounds like it's already decided. It's probable, but probability has to do with medians and means. The unique organism is not a median and mean or either one, either or that is, because why? Because it's unique, right? And it's not part, it may be you may be able to add it into a population, but the individual is not a population. It's not a universal. It's a specific. So we age and we deteriorate and we die completely on our own. See? So this led me way back when, I don't know how long ago now, 10, 15 years at least, to come up with a die-event ontological theory of living systems. And with a theory like that, you have to have some kind of structure to be able to point to, and I call it the dieventome for short. Now, what the dieventome has is this trigonal planar organization, because that's the simplest kind of organization where I can have three things interacting together. Okay? So you have the genome, which is the inherited imprinted gene expression system, includes single nucleotide polymorphisms, splice variations, copy number variations. This is all at the genetic level. That's why it's a genome. And of course, inborn errors. Genome is going to possess that at the get-go, at the beginning. It's the blueprint, right? You inherit all of that imprinted gene expression. You also inherit some imprinting from the epigenome from one parent or another. Secondly, you have the environment. That's a changing of stress, both internal, so the body is constantly interacting with itself because self has to recognize self so the immune system doesn't destroy host lineages, right? So that's a good way to understand it, at least at the initial level. You have an internal stress, constantly fighting against the propensity to destroy cells or even rearrange them when they're not necessary, so not to go to autophagy when it's not necessary, and hence shut down for a while during repair processing the function. Right? And certainly you don't want to have premature programmed cell death, because that can destroy the entire function of an entire system. And when that happens in the central nervous system, well, we know what that looks like, right? So the external then is something that comes from the outside, and that involves pathogens, of which there are, of course, a whole host of possible candidates. Xenobiotics, likewise, similar kite. Diet, medical treatments themselves as you age, because many people in the 21st century are on some kind of medical pharmaceutical diet, right? Psychological stress, which really affects some, not all. And the more that it affects the individual, the more that's going to change the entire repertoire of the homeostasis of the system, right? Because it's going to involve neuroendocrine hormones, for example. And it's going to include an amount of the immune system that's going to permeate and promulgate those psychological stresses. Because those are biochemical phenomena, and those are pathophysiological phenomena, at least at some level. You also have neurological, which now I'm separating and bracketing off the psychology. Neurological, cardiovascular, and metabolic disease. Metabolic disease like metabolic syndrome, like, cardiovascular disease associated with diabetes. Components of metabolic disease can actually also involve just changes in bioenergetics in whole organ systems. Muscle, for example, comes to mind. And the movement towards sarcopenia, or that is a senescence of muscle cells as one ages. Because the organ system that is known as musculature plays a major and vital role in the aging process. Not just the central nervous system, right? Not just the intellect. Of course, wound healing also is generating an external alteration. Remember, we talked about macrophages, type 1, type 2. Type 2 being involved in wound healing, type 1 involved in stimulating some cellular degeneration at times. Okay. You also have neoplasia to deal with. What's that? Cancer. Right? Oncogenesis. Neoplasia means just the production of new tissue. So when that gets out of hand, when it's no longer controlled, then you get an oncogenic event, which can lead to a tumor, which can lead to metastatic environment, which can lead also to death. Because remember, tumorous cells are cells arising from a population of normal natural cells that do not carry that function any longer. So having a tumor means you have a cellular mass without the function. Think about the liver, for example, right? Hepatocellular carcinoma, a good one that we talked a lot about, if you recall that, or go back and listen to it. Okay. So you have cancer, and then of course the aging is superimposed on all of these processes and exacerbating them, if you want to look at that way. You also have the immunoepigenome, I call it. This is real-time response to and alterations of the immune surveillance and activation. That includes inflammation. That includes anergy. That means setting off or bracketing off, for example, T lymphocyte activity for a while, or sometimes forever, but still having the interaction of those T lymphocytes, even though they're anergic. You have immune memory, T and B cells in particular, with interactions with antigen-presenting cells all the time, and with tissues like endothelia, epithelia. And you have this, again, epigenetic writing, reading, and erasure, which is always going on. If you don't think it is, think about how you learn things. You're learning something new maybe right now, and you're probably going to remember it in 10 minutes. Maybe after that, who knows, right? How does that happen? That's a writing into the genome, right? It's not simply just enhancing a certain wiring phenomena in terms of axon terminals and exponentials. How does all that happen? It happens by the alteration of gene expression, slight changes and that all involves an epigenetic phenomenon. It's not simply that a pathway is turned up or down. A pathway is micro-altered all the time when new information is enhanced into the knowledge base. Right? And that includes both knowledge itself, that we consider something that isn't a mechanical, physical thing that we can hold, although it is associated with biochemical and physiological responses. But it's beyond that, right? All of those things play a role. And so that's what the epigenomic editing is like when you think about it. It's the knowledge base itself. So let's look at some detail here. Journal called the International Journal of Molecular Science, right there is the citation, tells us that there are essential roles for these sphingolipids. Remember, ceramide was a sphingolipid, especially sphingomyelin and ceramide in the skeletal and smooth muscle. Now, we normally think of sphingomyelin and ceramide, at least I do. Ceramide is signaling, sphingomyelin, right? Normally associated with what? Myelinated sheaths in the central nervous system. Yes. Okay. Demyelination diseases like multiple sclerosis have been well described, right? So anyway, ceramide accumulation in skeletal muscle. So I'm saying it's in the CNS, but it's also in skeletal and smooth muscle, like the endothelia. Ceramide accumulation in skeletal muscles have been detected in response to numerous nutritional and stressful stimuli, such as high circulating free fatty acid, insulin resistance, and oxidative damage. And those last three, those are straight up pathophysiological phenomenon of type 2 diabetes. High levels of circulating free fatty acid, insulin resistance, and oxidative damage. Okay. Now I'm telling you that you get ceramide accumulation in association with that. I'm not talking cause and effect here. Cause and effect you have to kind of throw out the window, because a feedback control and feed forward means to generate new routes for ultimately signaling back to pathways, biochemical pathways. Those new routes aren't something that are all generated de novo, of course, but the lack of linearity of individual pathways is something that can constantly be invoked and then removed, depending on the needs of the system. Remember, we're heterotrophic organisms living in a free environment, and we have free will. That means that nothing is perfectly determined. Things change, and the body, and therefore the human with its intellect, adapts. This is all something to do with biochemistry and physiology, obviously. But more than that, right? Beyond that. So it's an extremely complex system. There's no wonder when it starts to fade and degrade as we grow older, it's disturbing, right? Of course it is, very disturbing to see loved ones, for example, start to senesce in many ways, right? Okay, so, and this can include earlier maladaptations, such as obesity, which can generate type 2 diabetes, and then leading all those changes in skeletal muscle and endothelial muscle. You see? So ceramide increases in rodent soleus muscle, it's a certain type of muscle in the rodent, and that's subjected to acute and long-term hind limb suspension. So when people work with rodents, this is how they mimic sedentary lifestyle. And a sedentary lifestyle is unfortunately a behavior that's chosen by free will by the individual that is often linked to obesity, because obesity slows a person's movements down, makes it more difficult to get around, and therefore enhances a lethargy signal that is procured and is ultimately the result of choices the individual makes, of course. So all of this, though, has biochemical consequences, you see. The choices have biochemical consequences, not the other way around. So sphingomyelinase, the enzyme we were talking about a while back, remember the acid sphingomyelinase I talked about in the audio lectures recently and also in video. Sphingomyelinase can be neutral or acid, and that depends on the pH in which they're optimum. Sphingomyelinase mediates ceramide-enriched microdomains, which then merge spontaneously into large raft platforms, cholesterol ceramide rafts I talked about, may aggregate protease and other receptors within that raft from their biosynthesis through the ER and Golgi. And even the peroxisome for some fine tailoring of the membrane for sure. And you also get signaling molecules, thus that translates induction via acid sphingomyelinase activity, all of which can be induced by a pro-inflammatory cytokine called tumor necrosis factor alpha, TNF alpha. And you get a lot of TNF alpha from obesity, because that is a chronic oxidizing pathophysiology, which generates pro-inflammatory cytokines, such as TNF alpha. You see. Okay. This is how morbidity can lead to more morbidity, you see. All right. So let's get more into this. If sphingomyelin hydrolysis is induced in situ upon stimulation of the acid sphingomyelinase, there will be a discontinuity of two membrane systems. The dislocating and reorganizing raft system, and left behind reorganized, refused membrane, endomembrane, think about it. Okay. This will occur because of redistribution of mole percent of sphingolipids, such as sphingosine, ceramide, but also glycerolipids like phosphatidylcholine and prenolipid cholesterol, okay, as well as the reorganization of the proteins embedded there, with which annular lipid domains, the annular means something that makes a circular core around those proteins, or the proteins can be embedded on the lipid raft itself, right? You can see that both domains can be generated. And they maintain as bioactive resource centers these rafts for docking, hormonal binding, secretion, cellular uptake, endosomal and exosomal trafficking, cell membrane potential changes, and the entire range of importing and exporting of biofuel, such as fatty acid and glucose, from external to internal, internal, external, using, with the lipid anyway, lipoproteins, for example, or serum albumin. But for the glucose, just plain glucose, and glucose then being trafficked in via GLUT transporters typically. Thus, that is going to alter bioenergetics, this membrane raft. Oxygen tensions, obviously, because oxygen doesn't dissolve well in aqueous, and a range of possibilities that can lead to hypoxia, there you go, and autophagy on one end, and programmed cell death on the other. So you see the complexity here and how absolutely beautiful it is, right? So you have plasma membrane lipid asymmetry, and that is set up by the genome, and is absolutely necessary, and in fact it's universal for cellular integrity. You can find necessity and universality in physical structures. That doesn't mean that they're always there, though. Right? So it becomes almost a nominal event, yet it is a physiological necessity. All right. So pretreatment of muscle cells with better put my glasses on here. Clomipirene, which is a functional inhibitor of the acid sphingomyelinase, okay, which are known as phiasmas, right, can stabilize raft integrity in synaptic junctional domains. So if you treat a muscle cell with clomipirene, which is an inhibitor of the acid sphingomyelinase, you can stabilize raft integrity because you slow down the production of ceramide this way, right? Because you inhibited the enzyme responsible for ceramide synthesis, at least in the acidic compartment, like an autosome or autophagolysosome. And that will then give you more integrity in the junctional domain of the whatever that membrane system is, such as a neuronal synapse. Recall, because I mentioned it before, that serotonin reuptake inhibitors, selective serotonin reuptake inhibitors, such as fluoxetine, which is prescribed after diagnosis of major depressive disorder to millions and millions of people in this country and worldwide, tens of millions of people. Those drugs, SSRIs, are actually phiasmas. That's right. They are functional inhibitors of the acid sphingomyelinase. And that's their actual mode of action, what I just described there. They stabilize raft integrity in the synaptic junctional domains of neurons. And therefore, they act to inhibit serotonin reuptake because the serotonin reuptake requires membrane lipid raft mobility and reorganization. Hence, that's what these drugs do. How they actually do it is rather simple. They actually increase the pH of the autosome, endosome, autophagolysosomal compartment where the sphingomyelinase, acid sphingomyelinase happens, where it's active. If you raise the pH of a subcellular compartment where you have an enzyme that has an acidic pH optimum, like acid sphingomyelinase, what happens when you raise the pH? You shut off the activity of the enzyme. Shut off the activity of the enzyme, decrease the amount of ceramide production. You change the functional domain of those membranes around that region where that acid sphingomyelinase has been inhibited by this SSRI, which is a phiasma, and you get serotonin reuptake inhibition. So the serotonin continues to work, and there is some suggestion that serotonin, with constantly agonizing through that synapse, is functional for decreasing the action potential transmission in certain neurons, thus affecting ultimately major depressive disorder. It's a mode of action through alkalization of a compartment containing an acid sphingomyelinase. Recall, after saying all that aggregation of membrane proteases are linked to coronavirus infection, and that aggregation is induced by a ceramide-mediated lipid raft insertion into damaged or otherwise diseased plasma membranes, disease meaning corrupted in some way. Okay. Because the receptors, the ACE receptor, for example, for that coronavirus, right, the angiotensin converting enzyme 2, ACE2, is moved by membrane lipid rafts enriched by acid sphingomyelinase produced ceramide. Don't you know? See how this thing links together, okay? Now that's just an aside. I'm not talking about coronavirus in this lecture. Just something that you find out. Now it's just showing you some actual data. Whoops, sorry. Where did we go? Let's go back here. Yeah. How did that happen? I don't know. You use a tricyclic antidepressant like citalopram, or you use Lexapro or fluoxetine like Prozac, Paxil, which is paroxetine, or sertraline or Zoloft. These are all drugs that act like phiasmas, right? They're affecting the acid sphingomyelinase activity. So you can see this is looking at ceramide fluorescence in this tissue. Control, 12 hours, and 12 hours plus adding one of these phiasma drugs. Ceramide fluorescence increases upon muscle damage. That's the fluorescence there. And this effect is knocked back down after clomipramine, which is a phiasma drug. In fact, clomipramine is a tricyclic antidepressant, some of the old school antidepressants, right? They still work the same way, right? They're affecting the acid sphingomyelinase. And so you see ceramide levels start to drop. You see ceramide levels start to drop. Now, this is being discussed and examined here in this particular paper. You see, this is the histogram showing you the decrease in ceramide fluorescence. Control, after giving that the muscle to be held down, which is an animal model for enhancing depression in the animal, major depression disorder model. Right? You increase the amount of ceramide, right? And now what you're doing is you're removing that ceramide activity because you've decreased, in this particular muscular system, you've decreased the amount of ceramide production because you made alkaline that endosomal compartment where the ceramide would be generated. Now, this is a muscle tissue readout, but you can understand the same thing happens at the synaptic membrane level in neurotransmission, presumably in humans. And it's more than presumably, it's been well documented. It's the mode of action of these drugs. So, SSRIs as well as tricyclic antidepressants. You understand? Yeah. The reason these things are looked at, first of all, we need to know about these cationic amphipathic drugs, how they function. But we also want to know if they could be used in pharmacokinetic and pharmacodynamic experiments to study things like muscle damage and sarcopenia and aging, but also to study, ultimately, sure, neuropsychiatric diseases like major depressive disorder or general anxiety disorder or bipolar disorder, right? Of course. So, cationic amphipathic drugs. Now, the paper that we're talking about here was published in 2017, so it's 4 years old. Gave us new insights into the intracellular distribution pattern of cationic amphiphilic drugs, which is what those are. You can see the citation there. So let's go through this. This is basically just an abstract, but it gives you more detail. And that's what I want to do here. CADs, cationic amphiphilic drugs. Amphiphilic means that they can both interact hydrophobically and hydrophilically. Comprise a wide, like tricyclics, comprise a wide variety, which means they can move through membranes. Sorry, more organic chemistry there. Comprise a wide variety of different substance classes, such as antidepressants, antipsychotics, and antiarrhythmics, okay? Such as used for seizures, right? It is well recognized that CADs accumulate in certain intracellular compartments, leading to specific morphological changes to the cells. So far, no adequate technique existed to this point, allowing for ultrastructural analysis of CAD intact cells. Azidobutramine, a recently described multifunctional antidepressive analog, I remember when this came out, allows for the first time to perform high-resolution studies of CADs on distribution pattern morphological changes in intact cells. They show here, again, this is an abstract, so I'm quoting, that the intracellular distribution pattern of azidobutramine strongly depends on drug concentration, of course, concentration dependent, and exposure time. Both those things are very common for pharmacological studies. The mitochondrial compartment and the late endolysosomal compartment, and those are showing you, they know that that's what they are because they have these particular proteins embedded in them. That's how they scan through and understand that's what they're looking at. Were preferred localization sites at low to intermediate concentrations. That's down at the micromolar level. In contrast, they found the autophagosomal or phagolysosomal compartment, shown with LC3, with the green fluorescent protein binding to LC3 protein, can only be reached at high concentrations, that is above, at least approaching 10 micromolar. So 1 to 10 times the concentration that you'd find up in those, in the late endosomal lysosomal or the mitochondrial department, right? So it's now you need higher concentration to get into finally that late phagolysosomal compartment, excuse me. So at the morphological level, LC3 clustering became only prominent at high concentrations, while changes in CD63, which is the surface protein, pattern already occurred at intermediate concentrations. So they say, to their knowledge, it's the first study to establish a link between intracellular CAD, that's the drug, distribution pattern, and morphological changes. Therewith, Their results allow for gaining a deeper understanding of intracellular effects of cats. So this gets down to just studying basically the chemistry of these drugs, but showing you that you increase concentration, you change where those drugs end up inside the cell until you get to the compartment, the autophagolysosome, for example, where acid sphingomyelinase is resident, thus altering the pH of that environment, thus altering membrane RAF ceramide levels, thus altering neurotransmission because of changes in the fluctuation of the concentration of the neurotransmitter serotonin. You see? Yeah. That's where we're at with it. Now, the reason I'm going through all this detail is because this is authentic biochemistry. What do you want, right? And also so that you understand that a mode of action of a drug, like saying, well, it's a selective serotonin reuptake inhibitor, that doesn't really tell you at all about the mode of action. Here I'm explaining to you what the mode of action is down to the chemical level, right? Because this is what's important. So any drug that would alter the alkalinization of an endosomal compartment could have a very similar effect that something like as sophisticated as an SSRI or a tricyclic antidepressant might have. See? And what it might also do is alter, because it's altering membrane lipid raft, ceramide to cholesterol ratios, change the movement of proteins to the surface of the plasma membrane. What kind of proteins? Surface receptors. What kind of receptors? Receptors to docking to, say, ligands that include virion. Right. Now you see. So this just gives you more detail here. The LC3 lipoprotein strongly linked to autophagy, and autophagy-associated processes. Furthermore, it's well known that CADs modulate autophagy and autophagy-associated processes, and they intervene with that LC3 and LC3 metabolism, which means turnover. They show that the co-localization of LC3 and azidobutramine occurred only at concentrations of 10 micromolar. Again, that's getting high enough to get into the autophagolysosome. And exposure times of at least 24 hours. It takes a while to then titer up, right, that effect of the drug on the system. It is not clear at this point whether this co-localization of physiological relevance, we know that it is now, or a mere reflection of the massive intracellular overload of azidobutramine. Okay, so that's what they were saying way back when these papers were published. Okay? So you get the idea how earlier publications build on current publications, which become the knowledge base necessary to evaluate pathobiochemical and pathophysiological phenomena, which is what we're doing here when we're discussing aging. Okay, so this is the level of granularity. I don't really like that word, but we'll go ahead with it. I like to call molecularity. How's that? The level of molecularity to be able to fully describe to you what the aging process is, and then go all the way from there to discuss things like neuropsychiatric disorders, muscle sarcopenia, endothelial degradation, neurodegeneration. You see? These are all phenomena that are linked way back to the level of molecular interactions, all superimposed upon the way the organism, in this case the human, lives its entire life, going all the way from conception, that's where we started a couple of days ago, to the last stages of morbidity and mortality, and finally death. Choices made, changes in differentiation and development early on, but choices made once the organism is generating and exercising their free will. Choices in diet, choices in environment, choices in interaction, choices in how the individual deals with interactions, all will affect biochemical and physiological phenomena, not the other way around. That's the whole point of this lecture, the whole point of discussing aging. Aging is what we do to ourselves, basically, and as is everything else, right? Not just the aging process. Okay. So I'm going to leave you with this last bit of writing here because I know we've been doing a lot of intense discussion here. I don't want to keep on going on and on and on because I have a lot to talk about, right? You can see why I do a lot of this audio because I can just read it to you, right? And then as I'm reading it to you, I can explain it like I'm doing now. But we can move faster because now I realize I'm on camera and I want to look at you and explain to you and to emphasize things. So I think it's useful. It would be much better if we were in a lecture hall, trust me. So let's go through all this so we can finish for today's video lecture, okay? Several sphingosine-based bioactive lipids are found within membranes or trafficking through them, and they have been implicated in normal and pathophysiological states, and they are actually regulatory signaling molecules, not just membrane components. The whole thing with ceramide, basically it looked like it was just an inert biophysical phenomenon moving around membrane rafts, right? Which is what it was doing. But I'm telling you, they also signal. So they have different modes of interaction in biochemical and physiological responses. That's the point. So besides sphingomyelin, the major lipid found in the myelin sheath of critical neuronal tracts in the central nervous system, for example, the associated metabolite ceramide, ceramide 1-phosphate, just simply adding a phosphate to it covalently, which can be removed, by the way, sphingosine and sphingosine 1-phosphate, which are intermediates, degradation and biosynthesis intermediates for ceramide and sphingomyelin, by the way. And I went through that pathway several times before, and I'll do it again. They're all necessary bioactive sphingolipids, all within an interactome, all within a membrane of, say, an important neuronal synapse, you see. The role of cerebrosides, which we also talk a lot about in the central nervous system, such as the galactosyl, glucosyl, sialyl, and sulfonyl sphingolipids, is still not absolutely clear. We know that they are important for membrane plasticity and bioactivity because of the fatty acids involved, usually very long chain polyunsaturated fatty acids of the omega-3 series, such things as icosapentaenoic and docosahexaenoic acid and docosapentaenoic acid omega-3 type. We know that those fatty acids and those lipids act both as a surface control over electrical dissipation, right? That's one of the things that sphingomyelin does in myelinated axons. And we talk about that saltation of neurotransmission, for example. But there's something else they're doing because they're constantly being interconverted. So they're signaling as well and dealing with things like the ionic properties of ligands and receptors, which is what we just went through. Right? So cerebrosides are extremely complex. We know this because of inborn errors of metabolism. When we don't degrade or turn over cerebrosides, we have severe problems with mentation in very young children, lack of development, and often early death. Those inborn errors, they're just dreadful. Okay, so these cellular and extracellular trafficking sphingomyelin sphingolipids are interconvertible via enzymatic pathways that are not always expressed constitutively in any particular cell type. They have to be induced, right? They are implicated, however, in various physiological phenomena, including inflammation, which we've been talking about off and on for months, stress resistance, proliferation, differentiation, and in the plastic and elastic remodeling of neuronal and glial cell fate in the CNS. Glial cell like microglia, for example, which act as resident macrophages. And yes, they can exist in an M1, M2 component, like I've been talking. Pro-inflammatory versus anti-inflammatory, right? These potentiating sphingolipids are implicated in cell division, growth, apoptosis, autophagy. These are all mechanisms. These are all mechanistic alterations in cellular and tissue responses. Senescence, adhesion, migration, inflammation, angiogenesis, laying down new vascular beds, and endosomal plus organellar intracellular trafficking, which we were just touching upon with peroxisophagy, and mitophagy, for example. Much more detail there too in the mitophagy, which we'll do later. Opposing or reinforcing modes of action have been ascribed to individual sphingolipid classes and molecular-specific subclasses. For example, ceramide is a harbinger of programmed cell death. That's been well described, while phosphorylating ceramide is C1 phosphate, ceramide 1 phosphate, or even sphingosine 1 phosphate, sustains cell viability and transformational viability, which means because the cell is vital, it can also go through a genetic or epigenetic transformation and then become oncogenic. You see, so programmed cell death seems bad because it's degeneration, but if you don't have a quick way to get to degeneration, you can get to proliferation. If you get proliferation, you have a very sick cell, very sick cellular system, tissue system, and ultimately you can generate tumors, right? Mere addition or removal of ATP derived anhydride phosphate as an ester to preform polypeptides can reverse the direction of signaling. We talk about this all the time, kinase versus phosphatase, and change in description or translation in participating molecules, thus providing a molecular circuitry network to mediate cell fate. And we talk about this just in basic biochemistry. Now you're getting authentic biochemistry, you see. It's therefore not a surprise to biochemists, and hopefully to anyone that learns this, that bioactive sphingolipids will be involved in the initial and closing arguments toward aging and neurodegenerative disease. I'm going to leave you with that idea, okay, now. So we can close off this lecture, which I hope was as much fun to hear as it was to give, because what I wanted to do here, what I wanted to do here was give you what I really want to emphasize with the aging arc that we've been doing ever since last fall, right? It's fall again. I told you I was going to finish this in the fall. I told you before fall, well, I'm like pretty far along right now. These are synoptic lectures. I would like to be able to put in all of the bells and whistles that I put into all those series of lectures. I don't know how many there were, 60, 70 lectures easily. I never really go back and count them, but I know there's probably that much material there. And I can't do that. Then I would just be reiterating all those lectures. So I'm trying to select from those lectures and adding constantly new material, because that's what I do, because I want to keep way up front, in fact, ahead of the curve in understanding what's going on in the published literature. Hence, that's what I do all the time, so that we are aware of whatever comes around the bend, right? But having a full, comprehensive knowledge of everything that pre-existed in terms of publication is exactly impossible. What I can do is use my expertise to go into that literature and constantly, like the immune system, surveil and act upon it and bring it out in a form that can be interacted with and networked so that we gain a knowledge base in a particular system we want to study. And the one that we're trying to finish off here is the aging system, right, in humans. So with all of that in mind, I think I'm ready to close off our lecture. So I'm going to do that, saying this is Dr. Dan Guerra on the 22nd of September, the very first day of fall, my favorite season, autumn, and saying bye for now.